Welcome to today's lecture on the intricate topic of membrane potential and action potentials. As medical students, it is crucial to understand the fundamental principles underlying these electrical phenomena, as they form the basis of neurological and cardiovascular physiology. Throughout this script, we will delve into the different aspects of resting membrane potential, action potentials, and their modulation in both health and disease. Additionally, we will explore the role of ion channels, genetic mutations, pharmacology, electrical synapses, muscle contraction, computational modeling, cardiac activity, neuromodulation techniques, and their impact on these electrical signals. Let's get started with the first topic, resting membrane potential. The resting membrane potential, RMP, is the baseline electrical potential difference across the plasma membrane of a neuron or muscle cell when it is at rest. It is crucial for the transmission of electrical signals and maintaining cell homeostasis. The RMP is typically -70 mV, MV, in neurons. This negative potential is primarily contributed by the relative concentrations of ions inside and outside the cell. The cell's plasma membrane is selectively permeable, allowing certain ions to travel across through ion channels. Potassium ions, K+, play a significant role in establishing the RMP. At rest, the concentration of K+ is higher inside the cell than outside. This concentration gradient drives K+ efflux through leaky potassium channels in the plasma membrane. This efflux creates an excess of negatively charged ions within the cell, leading to the negative RMP. In addition to potassium, sodium ions, Na+, and chloride ions, Cl, also influence the resting potential. However, the plasma membrane's selective permeability to these ions, along with the activity of ion pumps, maintains their concentration gradients, resulting in a net negative potential across the cell membrane at rest. Action potentials are transient changes in membrane potential that allow for long-distance communication within the nervous system. They involve depolarization, repolarization, and hyperpolarization. Depolarization occurs when a stimulus increases the membrane's permeability to sodium ions, causing an influx and upstroke. This triggers the opening of voltage-gated sodium channels, leading to further depolarization and initiation of an action potential. Repolarization is caused by potassium ion efflux, restoring the negative membrane potential. Hyperpolarization briefly occurs before returning to the resting membrane potential. The intricate mechanisms of the resting membrane potential and action potentials are just the tip of the iceberg. For those interested in specialized and advanced study, there are several fascinating areas to explore further. One such topic is the role of ion channels in generating and modulating action potentials. Different types of ion channels, including voltage-gated channels, ligand-gated channels, and mechanically gated channels, contribute to the formation of action potentials. Understanding their properties and regulation can provide insights into neuronal excitability and the integration of signals within neural networks. Another area of interest is channelopathies, which refer to genetic disorders resulting from dysfunction in ion channels. These disorders can profoundly impact membrane potential and action potentials, leading to various neurological conditions such as epilepsy, ataxia, and channelopathies-induced cardiac arrhythmias. Exploring the underlying mechanisms and potential therapeutic targets of channelopathies is a fascinating and clinically significant field. Additionally, the pharmacology of drugs targeting ion channels and action potentials is of significant importance in the treatment of several disorders. Understanding how these drugs modulate membrane potentials provides valuable insights into their therapeutic mechanisms and potential side effects. Channelopathies are genetic disorders characterized by dysfunction in ion channels. These disorders can have a profound impact on membrane potential and action potentials, leading to various clinical manifestations. For instance, mutations in voltage-gated sodium channels can result in hyperexcitability of neurons, leading to conditions such as epilepsy. In contrast, mutations affecting potassium channels can cause reduced excitability, potentially leading to muscle weakness or cardiac arrhythmias. Understanding the specific ion channels affected in different channelopathies is essential for accurate diagnosis and appropriate management. Furthermore, studying these channelopathies can provide valuable insights into the physiology and function of ion channels in normal and diseased states. The generation and propagation of action potentials can be modulated by various neurotransmitters and hormones in the body. These signaling molecules exert their effects by targeting specific receptors on the cell membrane, leading to changes in ion channel activity and membrane potential. For example, the neurotransmitter acetylcholine can open ligand-gated ion channels on postsynaptic neurons, resulting in membrane depolarization and increased neuronal excitability. In contrast, hormones such as cortisol can influence ion channel activity through indirect mechanisms, affecting gene expression and protein synthesis. Understanding the intricate mechanisms by which neurotransmitters and hormones modulate action potentials is crucial for comprehending the complexity of neuronal communication and the regulation of physiological processes. Genetic mutations in ion channels have been linked to numerous neurological disorders. These mutations can alter the properties of ion channels, affecting membrane potential and action potential generation. For example, mutations in the voltage-gated sodium channel SCN1A have been associated with Dravet syndrome, a severe form of epilepsy characterized by recurrent seizures. Similarly, mutations in potassium channels can lead to channelopathies such as Andersen-Awell syndrome, which is characterized by muscle weakness, periodic paralysis, and cardiac arrhythmias. Studying the relationship between genetic mutations, ion channels, and neurological disorders is crucial for elucidating disease mechanisms and developing targeted therapies. Pharmacological agents targeting ion channels and action potentials play a significant role in the management of various disorders. These drugs can modulate ion channel activity, either by enhancing or inhibiting their function, thereby influencing membrane potential and action potential generation. For instance, anti-epileptic drugs such as carbamazepine and phenytoin act by inhibiting voltage-gated sodium channels, reducing neuronal excitability and preventing the generation of excessive action potentials. Understanding the pharmacology of these drugs is essential for their optimal and safe use, as well as for developing novel therapeutic strategies for conditions involving ion channel dysfunction. In addition to chemical synapses, electrical synapses play a crucial role in the propagation of action potentials between neurons. Electrical synapses are formed by gap junctions, which allow direct electrical communication between cells. Rather than relying on chemical neurotransmitters, electrical synapses enable rapid and synchronized transmission of electrical signals. This property is particularly crucial for certain types of neural circuits, such as those responsible for reflexes or coordinated muscle movements. Studying the properties and functional significance of electrical synapses provides valuable insights into the complex network dynamics of the nervous system and its role in behavior and physiology. Action potentials also play a vital role in muscle contraction and the process known as excitation-contraction coupling. In skeletal and cardiac muscle cells, action potentials are responsible for initiating the sequential events leading to muscle fiber contraction. During excitation-contraction coupling, an action potential propagates along the muscle cell membrane, leading to the release of calcium ions from the sarcoplasmic reticulum. These calcium ions then bind to specific proteins within the muscle cells, initiating the sliding of actin and myosin filaments, resulting in muscle contraction. Understanding the intricacies of excitation-contraction coupling and the role of action potentials in muscle contraction is crucial for comprehending muscular physiology and the pathogenesis of muscle-related disorders. Computational modeling has emerged as a powerful tool for investigating action potentials in neural networks. By developing mathematical models that simulate the biophysical properties of ion channels and the electrical properties of neurons, researchers can gain insights into the complex dynamics of the nervous system. Computational models allow the manipulation and visualization of various parameters, aiding in the discovery of underlying mechanisms and predicting the behavior of neural circuits under different conditions. This approach has enabled researchers to unravel the fundamental principles governing neuronal activity and synaptic integration, contributing to our understanding of normal brain function and neurological disorders. Action potentials are also responsible for the electrical signals in cardiac cells and the initiation of the heartbeat. In the heart, electrical signals propagate through specialized cells known as cardiomyocytes, allowing coordinated contraction of the atria and ventricles. The initiation of the heartbeat is regulated by a specialized cluster of cells called the sinoatrial SA node, which acts as the heart's natural pacemaker. The SA node generates regular action potentials, which spread to the atria and trigger the coordinated contraction that propels blood into the ventricles. Understanding the complex mechanisms underlying electrical signals in cardiac cells and the initiation of the heartbeat is crucial for diagnosing and managing cardiac arrhythmias and other heart-related disorders. Neuromodulation techniques, such as deep brain stimulation, DBS, have revolutionized the treatment of several neurological and psychiatric disorders. DBS involves the implantation of electrodes into specific brain regions, which deliver electrical impulses to modulate neuronal activity. By modulating action potentials and altering the activity of neural circuits, DBS can alleviate symptoms associated with conditions such as Parkinson's disease, essential tremor, and obsessive-compulsive disorder. Understanding the underlying mechanisms by which DBS impacts action potentials and neural activity provides valuable insights into the pathophysiology of these disorders and aids in the development of novel therapeutic strategies. In conclusion, the study of membrane potential and action potentials is important for medical professionals. It involves various complex topics such as resting membrane potential, action potential generation, channelopathies, neurotransmitter and hormonal modulation, genetic mutations, pharmacology, electrical synapses, muscle contraction, computational modeling, cardiac activity, and neuromodulation techniques. Understanding these mechanisms can help in diagnosing, treating, and managing different physiological and pathological conditions. I encourage you to delve deeper into these subjects and appreciate the intriguing realm of bioelectricity as you progress in your medical career.